I am investigating the cell biology of yeast. That's the simple one-sentence description of what we're doing. We're looking at some more specific things as well. So we're trying to analyze and understand a particular metabolic pathway that some yeast are able to carry out in the environment. And the same biochemical pathway can influence the flavor and aroma of beer and wine if the wrong yeast get into those drinks while the fermentation is taking place. There's both a simple basic science aspect to the project where we're simply trying to understand the physiology of the yeast, and then there's the interesting applied aspect to the same research. It's kind of a joining of long-time interest and hobby along with my experience and training in cell biology. My research in virology is still in the back of my mind, but funding has been a major problem, and so this is a line of research that uses many of the same resources and technologies, but is a much simpler and, quite frankly, less expensive system to work on. We're interested primarily in yeasts that are members of the genus Brettanomyces, which are very common in the environment worldwide and are the major cause of wine spoilage in that industry. So there's a lot of commercial application to this. But what we're really interested in determining is if there are differences in this metabolic pathway between one strain and another strain. Because the same genus of yeast are also desirable in certain specialty types of beer. So you get bad characteristics in wine, some good characteristics in beer by the same species of yeast. So what are the differences? Is there subtle differences in this pathway from one strain to the next that we can use as a predictive tool to determine this is going to be a good one, this is going to be a bad one, and try to come up with ways to analyze this early on in the process. When we're doing these strain analyses, we certainly are interested in whether or not a strain is appropriate for using in the brewing industry. There's really never an instance where a winemaker wants to have Brettanomyces in their wine, or at least it's extremely rare if there are. But brewing with Brettanomyces for beer is growing in popularity both among home brewers and craft brewers. So we're always on the lookout for strains that have characteristics that are appropriate for the brewing industry. And Eric Luman here at Green Room has used one of our strains to brew several batches of beer, and we're always on the lookout for new ones as well. So Eric is a biology major at UNF. He's kind of part-time at this point since he's really involved with the brewery. But as part of a senior research project, when he was still the head brewer at Seven Bridges, he approached me about doing a research project with the yeast at Seven Bridges in order to form the basis for this senior project. So we partnered up in that sense, and he kind of led that project, and we got a publication out of that work. And when he moved to Green Room, we maintained that kind of collaboration, and we've just kept it going from there.
The yeast has shown a lot of potential. It's been really nice, the results. I mean, there's a lot of commercial yeasts available out there already on the market, but none of them are local. So this is kind of our chance to pull in something local and say, this is from here. This beer is, really ties into the local community. You can't grow malt locally, and you can't, which is barley. You can't grow hops locally, but we can harvest yeast locally. So we can really tie in a local ingredient. A lot of our other beers tie in stuff like local citrus, local spices, local fruits. So it just gives us kind of another avenue to pull into it, to really tie that local aspect into it. And it's a lot of fun to see what these yeasts put off, be it good flavors or bad. And it's neat. The beers we've done with it have gone over really well. I mean, we've sold them all. People seem to really like them. That local tie really draws. It's still got to be a good product, but that local tie really is popular with people right now, and we love the idea of it. Try and source as much local as possible. So it really is right up our kind of alley to do stuff like that. And I went for biology because that's what I liked anyway, and I'm able to put kind of practical experience to it here. It's nice. It's very hands-on. I get to pull in, a lot of the biology, the chemistry, physics, all, in my day-to-day operations. I'm not really doing straight lab work, but, we're doing that on, day-to-day, and really get to see fun results with the beer and people drinking and enjoying it, and I enjoy it. So it's fun, it's neat. It's a good application of the sciences.
So the recent article that we just had published is really a preliminary characterization of about a dozen new yeast strains that we isolated from around Jacksonville and other parts, actually around the eastern U.S. And so we did basic physiological, genetic, and biochemical characterization of all these strains, really as a preliminary characterization. And then we also determined from that characterization that several of them were potentially appropriate for commercial brewing. And that is how we partnered up with Eric here at Green Room to use some of those strains in the brewery here.